whenever you learn about cholesterol, other things other than the function is performing and its structural aspects, you hear a lot about its relationship with different diseases, specifically the heart diseases. So, which means that its regulation, the regulation specifically of its synthesis, is of real importance. Um, so, as in the previous lecture, we have talked about different processes which are, or different reactions which are involved in the synthesis of the cholesterol. There, we specifically talked about a reaction which leads to the formation of mevalonate, and there we saw that HMG-CoA reductase, this process in which HMG-CoA is converted into mevalonate, a 6-carbon compound, this reaction is a very important regulatory step as far as the synthesis of cholesterol is concerned. HMG-CoA reductase, it is activated, or its processing production depends upon many factors. One of the very important factors is the concentration of the sterol, which leads to the formation of course, cholesterol. So, that's why we say it the sterol-dependent regulation, because a compound known as sterol regulatory element binding protein 2, it is actually regulated. Its activity is regulated by the sterol concentration, and its activity then in turn leads to the formation of the HMG-CoA reductase enzyme, which eventually lead to the synthesis of cholesterol. So, if we see here, this is SREBP2 protein along which makes a complex with the SCAP protein, and when the concentration of the sterol is low, it will give the signal to SREBP2, which will be then cleaved from the SCAP, released from the SCAP, and released from the Golgi apparatus, and move towards the nucleus and binds with the SRE, sterol regulatory element, from which this protein takes its name SREBP2, because it binds with the SRE. As a result of this, is a cis-acting element, SRE. It binds with the cis-acting element, and transcription, it leads to the transcription of the HMG-CoA reductase enzyme. So, in this way, SRE, which is located upstream of the gene which is involved in the, whose protein is involved in the synthesis of the cholesterol, it is activated by SREBP2, which leads to the synthesis of more and more of the HMG reductase enzyme. Likewise, when the concentration of sterol is much higher, it will inhibit the production of HMG-CoA reductase through a mechanism which is very interesting to understand, that SREBP2, I have told you, is bind with SCAP. Here, when the concentration of sterol is high, SCAP doesn't allow SREBP2 to be released. Instead, it lets it to be bound with the INSIG, another protein known as insulin-induced gene proteins. Keep in mind that you don't have to just learn by heart the name of, whenever we're talking about so many abbreviations. What is more important, you know the function of these proteins. What is more important, you must know that how they perform their function and what is the meaning of this function for the overall physiology of the body. So, then what happens, that SREBP2 remains bound with the in the Golgi apparatus, and it doesn't go to the nucleus, and hence the SRE is not bound by the SREBP2. There is no much transcription of the HMG-CoA reductase and no much synthesis of the cholesterol. So, this is sort of a feedback mechanism, or negative feedback mechanism, which regulate the synthesis of cholesterol. Another way of regulating the synthesis of cholesterol is that sterol sensing integral proteins, they lead to the ubiquitination, the degradation of the protein or the reductase enzyme, which is involved in the synthesis of cholesterol. So ubiquitination is another way. The degradation of the protein is another way in which you can regulate the product, the synthesis of a product. Actually, what again here happens is that INSIG, insulin-induced gene protein, this mediate the degradation of this protein. Here again, this is a feedback mechanism because it depends upon the concentration of sterol. More sterol concentration, more will be the degradation of the reductase enzyme, lesser will be the production of the cholesterol. And this is how the concentration of the product regulates its production. And cholesterol, of course, is not an exception. So there are different drugs which attack the enzyme which is involved in the synthesis of the cholesterol, specifically the reductase enzyme, HMG reductase. And as a result of this, there is a less and less production of cholesterol. For example, the enzyme, the drugs like statin drugs, what happened that they are structural analogs of the HMG-CoA enzyme, HMG-CoA. As a result of this, because they resemble the HMG-CoA, there is a competition between these two compounds, the drug and the HMG-CoA. And as a result of this is what we say, it leads to competitive inhibition. And as a result of this competitive inhibition, what will happen that the drug binds with the target proteins as compared to the HMG-CoA. And as a result of this, the target protein, of course, is the enzyme here. And it leads to the decreased production of cholesterol. This drug is used if you are dealing with diseases like hypercholesteremia. In hypercholesterolemia, what happens that you have excessive accumulation of cholesterol in your body because of its excessive synthesis or its reduction in its degradation. So to deal with it, you take different drugs, specifically the statin-based drugs like atorvastatin, flavastatin, levastatin, and so on and so forth. And these drugs act as competitive inhibitors, and they lead to the decrease in the production of the cholesterol. In this way, you see that understanding the pathways, understanding different ingredients, components of the pathway is how important that it's not only important to understand the overall metabolic pathway, but also it leads to many clinical innovations in the medical field. Innovations which leads to drugs, it opens different therapeutic windows.